So again, from past 2 days, I've been showing you the video of differences between archaea. Not exactly the difference. I can say I was explaining about archaea, bacteria, and viruses. So, of course, you all recall the characteristics of prokaryotes and eukaryotes, the difference between them, that they're single cell and multi-cell. One has nucleus in the cytoplasm, the other has DNA in the cytoplasm, and the other has DNA in the nucleus. Prokaryotes, DNA in the cytoplasm. Eukaryotes, DNA in the nucleus. When I say describe the characteristics of archaea, what exactly when it comes archaea? I told you what is the difference between archaea and bacteria. Archaea can live in
the archaea can live in the boiling water.
Don't say boiling water, just write extreme conditions. That was an example. When I say boiling water, it's an example. You must say they can live in extreme condition. Extreme means either very, very cold or very hot. They're found everywhere on earth, including extreme environments such as salt lakes. Example, what they have given you is salt lakes. Okay, that is extremely salty. When I say bacteria, they have DNA, they have proteins, a cell membrane, and a very, very strong outer wall. Again, they have DNA, they have proteins, they have cell membrane, and a strong outer wall. Now, coming to the production, how archaea and bacteria are produced. Here, in short, they said that they are reproduced by binary fission. You remember I told you about vegetative reproduction and budding and binary fission?
Yeah, spore.
Yeah, same way. So they reproduce by binary fission. Some bacteria can exchange genetic material before dividing. Before dividing itself, they change the material. And the two cells join to recombine nuclear material and they undergo binary fission. You know what? Before the cell divide, if the nucleus meet the material of the nucleus inside, they meet before the cell divide. We say that they recombine nuclear material and they undergo binary fission. The material inside the nucleus of both, they meet and they make a new cell that is called, or new bacteria, that is by the process of binary fission. Bacteria can also take DNA strands from their environment. Even from the environment, they can directly take. Some bacteria form endospores around DNA and ribosomes to survive harsh conditions. If they have to stay in harsh conditions, they form some spores, some spots around the DNA and ribosome. Again, with reading.
Miss, what does it mean harsh?
Harsh is again like extreme. Very salty is harsh condition. Very cold is harsh condition. Where normally other bacteria cannot survive. Now this binary fission, you cannot understand that well when it is written this way. I will try to show you a video again. But before that, we have studied about this. We studied about the characteristic of bacteria. Now, this is a very important diagram here. See how the nuclei come, they fuse, and they split. We tell them as a split personality. In a binary fission, a bacterial cell copies its DNA. The DNA then separates and the cell divides into two new cells. And then they fuse, they copy themselves, then they divide, and then they finally become two bacteria from this. Now we'll just read the steps and we can go and watch the video. The first step in bacterial reproduction is copying the cell's genetic information. You know that always we studied that line. Before cells divide, the DNA is copied. You remember that line?
Yeah.
The DNA copy itself before cell divide. So that's what. The first step in bacterial reproduction is copying the cell's genetic information. This loop is called a chromosome. You know, bacteria, they have a long strand of DNA, and that long strand is called a chromosome. And it is copied, they become two chromosomes. Then these two chromosomes, they separate with one on each side of the cell. Next, you have seen in the picture how they separate. Next, the cell membrane starts to grow inward, separating the two halves of the cell. Finally, a new cell wall forms separates the two new cells. At the end of binary fission, there are two identical bacterial cells, each with identical DNA. So did you get an idea how do they divide and then they form their own cell wall? I mean, not cell wall, sorry, cell wall is only in animal. How they form their own cell membrane?
Yes.
All right. Let us see here.
As you know, there are 3 domains that organisms can be divided into: archaea, bacteria, and eukarya. Bacteria are a domain made up of unicellular prokaryotes that usually have cell walls and usually reproduce by cell division. Bacteria are an amazingly diverse group that can live in many
We have seen all these kind of bacteria yesterday. Spiral shaped, rod shaped. Now we have to see how these bacteria are.
Think about the images you have
Before
seen
that,
and
just
what you have
answer that.
learned. How are bacteria and archaea different? Click all correct answers.
What are same, girls? I think their DNA sequence
Nice job! Archaea and bacteria have different DNA sequences.
are different. Their size, it doesn't matter much. Their cell wall structure, no idea. Anyway, let us click
There
at
is.
That's right. Bacteria have cell walls that contain a mesh-like sugar amino acid layer outside the cell membrane, while archaea do not.
so means arrangement of DNA in archaea and bacteria is different. Of course, then only that's why we are saying them different DNA is different. I'm sorry, archaea is different, and because their sequence of DNA, the arrangement of DNA cell is different. Okay, their cell wall structure, size again.
That's not right. Their sizes are very similar. You can
That
try
they
again.
have same.
Good work! Archaea and bacteria produce different enzymes.
So what is the difference here?
While bacteria and archaea are about the same size, they
their
differ
DNA.
greatly in their cell structure, cell chemistry, and genetic structure.
Understood?
Yes.
Bacteria are able to reproduce in an amazingly short amount of time. They are able to do this because they reproduce asexually by splitting in two. The process by which one cell divides into two cells of the same size in a single-cell organism is called binary fission. Click the play button to watch this process.
See how now that binary fission process that's showing you in a video. See here.
This video is sped up, but the actual rate of reproduction is still very fast compared with that of eukaryotes. Binary fission can take anywhere from 15 minutes to 16 hours depending upon the type of bacteria and the growing conditions.
Did you see how they are multiplying? They multiply very fast, but some bacteria, according to the condition, they take their own time. They say from 15 minutes to 16 hours.
Now let's look at what happens in each step of the binary fission process in bacteria. Click the dots on the line to observe the process. The DNA of bacterial cells is arranged on a single circular chromosome. Before binary fission begins, the chromosome in each bacterial cell is folded into a tight ball. At the start of binary fission, the cell makes a copy of its DNA. The two copies of the DNA separate and each becomes one chromosome. The two chromosomes begin to move to opposite ends of the cell. As the two chromosomes move apart, the cell membrane in the middle of the cell moves inward until the cell's sides meet. When the sides meet, the two parts separate. This is the beginning of one cell becoming two cells. Each of the two new cells has one chromosome. A cell wall forms over the two exposed parts of the cell membrane at the separation point. What was one bacterium is now two new bacteria, which will each grow to the normal size and separate again.
Did you understand more better here with the picture?
Yeah, Miss.
How they separate and they become two new bacteria. This all happened in 10 minutes or 15 minutes. And not single. Maybe like if you have 100 bacteria, they'll make 200 bacteria out of it in 15 minutes. They multiply very fast.
Yeah.
Now let's look at another process called conjugation.
Conjugation, Vateen, I will explain tomorrow. I hope they come tomorrow. Because this is a
Okay.
new thing I don't like to explain. Something is just from before. And this all